Hi, hello, welcome back to the another session of chromatography. This is Sarita again, Assistant Professor of Chemistry, Government Degree College for Women, Begumpet. So in our previous classes, we have seen the introduction of chromatography and two techniques on chromatography. Those were TLC, that is thin layer chromatography, and paper chromatography. Now today, let us discuss about column chromatography. So the previous methods, that is TLC and paper, they were planar chromatography because the adsorption or the separation was done only on the plane of the paper, or it may be on the plane of the glass plate. Now what about this column chromatography? Here what we do is the separation will take place in the column. As you can see in the picture, this is the column where the separation of the mixture will take place. So let us go in detail with this topic. Column chromatography. It was first discovered or introduced by the scientist D.T. Day in the year 1900. So, as in the case of all the chromatography techniques, even this column chromatography technique is used for the separation and purification of the components from a mixture. So, before this D.T. Day or after, the other scientists were also doing this chromatography technique. And as we know that it was the first scientist, that is Michael Sweat, who has done the separation of plant pigments by using calcium carbonate. So after the scientist D.T. Day, who has developed this technique, later on many other scientists have used this technique for the separation. Now this column chromatography, we have different types of column chromatography. Those are, first one is adsorption column chromatography. So adsorption. So as you know that it is adsorption technique or adsorption phenomenon which is taking place. So when the sample is loaded onto the that is the adsorbent or the stationary phase, first the sample gets adsorbed. So even here in the column, we will be filling the column with a stationary phase on which the adsorption is taking place. In adsorption column chromatography, the stationary phase is solid and the mobile phase is liquid. As in the case of TLC also. TLC also, it is an adsorption phenomenon where stationary phase is solid and the mobile phase is liquid. The other technique is partition column chromatography. Now partition takes place always between two different, that is mobile phase and the stationary phase, which are made up of liquids. Now here also the stationary phase as well as the mobile phase are liquids. So we can use liquid as the stationary phase. How we can use liquid? Now as we have seen in the paper chromatography also, partition takes place there. Why? Because on the paper we find small pores where water is being embedded in those pores. So even here also, the liquid whatever we are taking as a stationary phase has to be embedded or supported on a solid. So when we are taking both stationary phase and the mobile phase as liquids, the principle involved is partition column chromatography. The next type is gel column chromatography. Now in this, the name itself, it gives information about the column is being filled with the gel. So gel will act as a stationary phase where the separation can take place. The last one is ion exchange column chromatography. Again, in this case, the column is filled with ion exchange resins. So because of this resin, which are having ions embedded on it, it will help in the separation of the ions. So these are the four different types of column chromatography where it is dependent on the principle what is used in it. So it is adsorption column, partition column, gel column, and ion exchange column chromatography. Now in the next slide, you can see what is the difference between the adsorption and partition. So as I said, in adsorption, you can see this is the stationary phase, the blue colored, and these are the solute particles. Or we can say that these are the different components present in a mixture. So you can see it is getting adsorbed onto the surface of this stationary phase. So stationary phase is the solid here, okay, and if the adsorption is more onto the stationary phase, then that particular component will move very slow. So we have seen the same in TLC also. When the solute or any component is strongly adsorbed onto the stationary phase, then that particular component will travel slowly. So the same case is observed even in column. Now in partition chromatography, you can see these are the solute particles, okay, which are getting equilibrated between the stationary phase and the mobile phase. In this, you can see this is the cross section of the open tube, the column cross section it is, where this is the stationary phase, that is nothing but a liquid which is being bonded to some solid. Okay, so the liquid is acting as the stationary phase. Even the mobile phase is also a liquid or a gas. So this solute will be partitioned between the mobile phase and the stationary phase. So if it is more soluble in the liquid which is acting as a stationary phase, then they go and bind here. Okay, so you can see here solute dissolved in liquid phase which is bonded to the surface of the column. So solute, as here these are the components. Solute means the sample here. The sample components are dissolved in the liquid phase. So adsorption and partition chromatography. Now, as I said, these are different types of column chromatography. Now, in all these cases, you can see the mobile phase is liquid. Then what about the stationary phase? The stationary phase is different. As I said, in adsorption, it is solid. In partition, it is immiscible solvent, which is present on a solid matrix. So we need that support. Otherwise, we cannot take that solvent as a stationary phase in this. Then in ion exchange resin is used. Then gel chromatography, where the solvent is held in interstices of the polymeric substance. So here, what happens is we will be having small spaces in the gels. Okay, so that solvent will be fixed in the spaces between that gel. So that will act as a stationary phase. That we'll see in detail in gel chromatography. And the sample phase in all these cases is we are loading the sample in a solution form. Now, let us go in detail about the adsorption chromatography technique. Okay, so adsorption chromatography technique where mobile phase is liquid and stationary phase is the solid. Now, mobile phase, as we said, it is liquid. Now, this is very important where the solvent, that is the liquid, whatever we are taking, it is acting as a solvent. That means it is helping in the eluting the components which are present in the mixture. So this is the picture how it looks in the lab. Now this is the column here which is made up of glass, and then this is the dropping funnel where the solvent is loaded, that is the liquid mobile phase is being loaded, and from this dropping funnel the mobile phase is drawn into this column, and this column it is having a stopcock at the bottom where you can turn it off or on, and all the eluting components which are coming from this column, they will be collected in separate beakers or conical flasks. So here the mobile phase, it is acting as a solvent. It is also acting as a developing agent, because what it is doing, the solvent, when we are running the solvent or the column, it will go and interact with the sample. This is a sample which is present here, and it is helping in the separation of the mixture. Various components from the mixture are being separated. That is, we call it as developing. So it is acting as a developing agent. It is also acting as an eluting agent. So what it does is, after interacting with particular component, if that particular component is more soluble in the mobile phase, then that component will be first eluted out from the column. Suppose here you can see two colors are there, yellow and green. Now this green component is traveling fast when compared with the yellow component. That means the mobile phase, what it is doing, it is making this green component get solubilized in it, and then it is bringing down the column, and this green part is being eluted out first, and that we have to collect it in a conical flask. So what are the mobile phases that we can use in column chromatography are? So here, as we have seen for in all the other techniques also, we generally start with the non-polar solvents. Now even here you can see it is N-hexane, cyclohexane, carbon tetrachloride, benzene, toluene, trichloroethylene, diethyl ether, chloroform, and now we are increasing the polarity as we are moving down. So it is ethyl acetate, N-butanol, acetone, ethanol, methanol, water. So what we are doing is we are starting from non-polar solvents and then later on we are increasing the polarity of the solvents. So we can take the mixture also. As I said, even in the paper chromatography, a particular solvent has to be, or the mobile phase has to be first, that is checked by trial and error method. So after selecting that particular solvent system, then only we can go to this column chromatography. So first we have to select the solvent system using TLC. After that, we can use the same solvent system for this column. Then what is the stationary phase that is used? Stationary phase, as I said, it is solid. So the solid, whatever we are using as a stationary phase, it should have uniform shape. The shape should be uniform and even the size. It should have both uniform shape and size. And the size should range in the order of 60 to 200 microns in diameter. That is, we call it as a mesh size. The mesh size usually that we use in column is from 60 to 200 microns in diameter. And it should show high mechanical stability. What is mechanical stability? Because this stationary phase, what we are selecting, we have to fill that stationary phase in the column. After that, what we are doing, we are loading the sample, and then also after that we have to add the mobile phase also. So it should be stable. It should not get dissociated when the mobile phase is run down through the column. So the stationary phase, whatever we are selecting, it should have high mechanical stability. Then it should be chemically inert. That means it should not interact chemically with either the sample or the mobile phase. So it should be chemically inert. It should be colorless, inexpensive, and readily available. So generally, if it is colorless, then only we can actually visualize the separation of the components. And it should be inexpensive. That means it should be of a lesser cost, cheap cost, so that we can use it in multiple times. And it is readily available. It should be readily available in the market. So generally, we have a lot of adsorbents which are acting as the stationary phases. It should allow free flow of mobile phase. So that stationary phase, it should not be very rigid. That means it should have some spaces between them, intermolecular spaces it should have, so that the mobile phase can be flown through the column. It should be suitable for the separation of mixtures of various compounds. So this is again the important point. So whatever solid we are taking, it should allow the separation of the mixtures. So these are the points we have to keep in mind while selecting a station. Okay, the common adsorbents what we use for column chromatography are, so here the list is given. The list of these adsorbents are in the order of increasing adsorption power. So in this you can see that alumina is at the last, where its adsorption power is more when we compare with all the other adsorbents. So adsorbents such as sucrose, cellulose, starch, calcium carbonate, calcium sulfate, calcium phosphate, magnesium carbonate, calcium oxide, silica gel, charcoal, magnesium oxide, alumina. So these are the various adsorbents that we can use for column chromatography. So generally we use silica gel as the adsorbent in all the column chromatography techniques. Now let us see what are the characteristics of a column, how the column should be, because this is one of the most important feature in this chromatography. Now the column, this is the column as you can see in the picture. This column, it is made up of glass. Okay, and how the column, what are the dimensions it should have is, the ratio of length to diameter should be greater than 20:1. So the length, suppose if this is the length of the column, if it is having 20 centimeters and the diameter should be 1 meter. And actually the ratio, it should be greater than 20:1. And what we take in as a standard is generally 40:1 as the dimensions of a column. And this is generally used in the lab. That is, we use the column which is 20 to 30 centimeters long and 2 to 3 centimeters in diameter. Now this column, suppose if the column is not available, it is nothing but it looks like the burette, what we see in our laboratory. So here you can see it is a long tube actually, which is made up of glass, and it is having a stopcock, same as that of the burette at the bottom of the column. So we can turn it off or on, so that when you want to collect the solvent from the column, then we have to put it on, or else you can just off the knob. Now after selecting a column, now what we have to do is we have to fill the column, because the adsorbents what we are using, we have to fill it in the column. So for the filling of the column, we have two techniques. One is dry packing or dry filling. The other one is wet packing or wet filling. So what we have to do is, first, when we take a column, we have to first wash it and make it dry. After that, you have to insert a glass wool. Suppose in this diagram, you can see, we need to insert a little bit of glass wool from the top. From here, from the top, we have to insert the glass wool. With a long rod, you can just insert it at the bottom of the column. After inserting the glass wool, now we have to fill it with the stationary phase, that is our adsorbent. Now the filling of this adsorbent is by dry or wet packing. So what we have to do is, in dry packing, we directly take the powder. Suppose if we are selecting the adsorbent as silica gel G, we have to take that powder directly and then start filling into the column. So let us see how the filling is taking place here. Now, as I said, there are two types. In dry packing, generally there are some demerits where we can see some air bubbles. So while filling the column, air bubbles can be entrapped between the stationary phase and the mobile phase. So that is the main drawback of dry packing. Or else some cracks can appear in the adsorbent layer. So these two we can generally observe while we fill the column with the dry packing method. Whereas in wet packing, what we do is we make a slurry of the adsorbent with the solvent, and then we fill the column. See, now here, this is the dry method, what I am showing now. So what we have to do is, so we are placing a glass wool first, and then sometimes there are we can also add some sand particles onto the glass wool, and then add silica powder. Directly the powder has to be added into the column from the top. Okay, so sometimes what we can do is we can also apply vacuum at the bottom of this column. So when you apply the vacuum, whatever the spaces between the particles of the adsorbents are there, they come closer. That is, the intermolecular spaces will be minimized. So when you are applying some vacuum, so you can see the level of the that is the adsorbent or the silica gel is still here, but when we apply the vacuum, you can see the decrease in the level. Now that means we say that it has been settled. The silica gel is settled in the column. Now next what we have to do is we have to run the solvent. So whatever the solvent we are taking, generally we start with non-polar solvents such as benzene. So take that solvent, benzene, chloroform, anything, then just keep on adding from the top. So this solvent will go and moisturize the silica gel which is present in the column. So until what we have to do is we have to open the knob also there. So when you open the knob, the solvent will, from the top as we are adding, it will go down till the bottom of the column, and then it will come out from the nozzle also. So we have to do this just for a while, such that all the silica which is present is getting moistened. Okay, it should become wet, or it should become saturated with the solvent. So after just checking, observing that all the silica has been saturated, now what we have to do is we have to just turn off the knob here. So when you turn off the knob, and you have to see that some of the silica, till here this is the silica layer, so still there is the gap, that is you have to fill it with a solvent so that this silica will not get dried up. So you have to take care that always the silica layer, upon that silica layer, you should have 1 to 2 centimeters layer of this solvent also. Because if you just remove complete solvent, what happens is the upper part of the silica layer will get dried up. So we have to see that the silica layer is moistened with the solvent layer for just 2 centimeters like that. So this is how the dry packing is done. Now there are some, as I said, there are demerits with this dry method. So what we have is, when you are adding the solvents, okay, sometimes you can see some of the air bubbles which are present in the silica layer. So these air bubbles will cause, that is the separation will not be effective when these air bubbles are present in the column. And also cracks may appear because at some part the silica get moistened and some parts it may be dry. That we don't know because at the center it is not visible at the center of the column whether complete silica has been moistened or not. So that is the main drawback of using dry packing method. Now we'll see the wet packing method. Now in wet packing, what we have to do is, so again we have to first insert the glass wool from the top into the column. Now what we have to do is just take the solvent, whatever solvent you are taking, just pour it 1/3 of that column. After that, you have to prepare the slurry. Slurry is nothing but where, suppose if you are taking silica gel, okay, we have to take the solvent and add this silica gel into the solvent so that you will get an uniform slurry is formed. Okay, so what we are doing, you take some solvent in the beaker and to that beaker add silica powder and you just take a rod and shake it or stir it properly so that it should be even slurry or uniform slurry consistency you have to get. Now after preparing that slurry, what we have to do is we have to pour that slurry into the column. So you can see this is the slurry which is being poured into the column. Now we have to tap in between. Suppose because the slurry, what happens is first we have added the solvent also. So sometimes the slurry may be, it may not go and settle at the bottom. So for that you need to tap the column so that after some time you can see that the slurry will get settled at the bottom. You can see this is the level of the slurry which has been settled. And on the top again you can see the solvent layer. Okay, so this solvent layer again you have to ensure that it will be on the top of the slurry again, because if it is not there, again the slurry may get dried up. Now you have to close the knob, and you have to see that the silica gel is covered with the little amount of the solvent there. So this is about the wet packing. Now in this case, we don't come across any air bubbles or cracks because already we are taking the adsorbent in a slurry form and then we are filling the column. So this is about the wet packing method. Now after the packing of the column, now what we have to do is we have to load the sample. Now here you can see how the analyte is getting adsorbed onto the silica. So as I said, the silica is polar in nature, which is having OH groups which are exposed here, you can see. Then this is the analyte or the sample molecule. Now this sample molecule, when it is loaded, it will go and bind to the polar OH groups of the silica. That means, suppose if this is polar, analyte is polar, it will go and bind strongly to this silica group, silica gel. Now when it is binding strongly to the silica gel, then when the affinity of this component is more onto the stationary phase, the movement will be very slow, because when we run the solvent, that is which is acting as the mobile phase, so what the solvent has to do is it should go and bind to the sunlight and then bring it down. But what happens is, as the sunlight is more strongly bonded to the OH group, it cannot bring it down fast. So that is why that particular analyte will travel at a slower rate. So it is nothing but the same principle again. When the analyte or the sample is strongly binded to the stationary phase, that particular analyte will travel at a slower rate. Okay. Now, these are the different steps in column chromatography. So now after filling the column, whether it may be dry or wet packing method, now what we have to do is we have to load the sample. So how the sample has to be loaded, it is again in the form of solution only. So now here the sample, what we load is should be concentrated. So it should be more concentrated and we have to apply in the form of a narrow band. Okay, we should not apply it as a broader one. So just a narrow band we have to apply, and then the same thing from the top we will be adding the mobile phase. So this pink color you can see now the mobile phase is being added or the solvent is being run down. Now that solvent will come in contact with the sample. Okay, and then after some time you can see the components which are separating out. So you can see two bands here. One is green, the other one is brown colored. So green colored band is traveling at a faster rate along with the mobile phase. So why it is traveling at a faster rate is because the interactions, it is showing weaker interactions with the stationary phase. Because it is bonded weakly to the adsorbent, it is traveling faster. Whereas this band is having stronger interactions with the adsorbent. So that is why it is traveling a bit slow. Now, as you can see, the traveling of this green band and it has come up to the bottom. Now you have to collect this. That is the component which is coming out or which is eluted out should be collected in a separate beaker. Then after some time, again you have to collect the another component, that is again the mobile phase, which will be coming out in another beaker. So series of beakers or the conical flask, we have to keep it ready and we should keep on collecting the fractions. So after that, again the other component will be eluted out. That should also be collected in another beaker. So the components with lower adsorption and affinity to the stationary phase, they travel faster when compared to the greater adsorption and affinity with the stationary phase. So the components that move fast are removed first, whereas the components that move slow are eluted out at the last. So now again in this picture you can see it clearly. Here this is the sample, mobile phase is added, molecules are separating. So further separation. So this is the red band. So each and every, whatever the mobile phase from the starting, whatever you are adding, it will come down and then that you have to collect it. Okay. So this is one fraction. This is another fraction after some time. This is another. You can see the green band moving down. So when the green band completely comes at the bottom of the column, now you have to collect that in another conical flask. Now complete red band, red colored component you will be collecting in a conical flask. Then next again the fraction without the molecules. That means this will be only again the mobile phase. So again the mobile phase will be collected in another conical flask. Then you can see the blue band moving down. So that blue band or the blue component which is moving down has to be collected in another conical flask. So this is how a series of fractions we have to collect. And what we have to do after this is we have to detect the components. So for that again we have to perform TLC. So we have to analyze the components by TLC. So as we can see the colored component, what we have to do is we have to take the sample and do TLC. So TLC, as you know, we have to first prepare the slurry and then we have to take the slides, glass slides, dip those slides into the slurry so that the adsorbent will form a uniform layer on the glass slide. Then you have to take a spot, that is sample of this by using capillary tube and load the sample on the glass plate. So when you load the sample and then take a solvent, put it in the that is TLC chamber, and then you have to run down the same procedure as that of TLC. Everything you have to do for this component. And you have to check whether a single spot is being observed or not. If you are observing a single spot, then we can say that whatever the component we have separated is pure. If it is giving rise to two or one more than one spot, then we can say that it is not pure, but it is having some impurity in it. So same as for the blue component, for this sample also you have to run TLC and then check whether it is pure or not. So this is how we do the column chromatography. Now this is the experimental thing what we use in the lab. So this is the column which is having a nozzle and a stopcock. So all the this one that is the conical flask are being kept ready. The column is filled with the adsorbent, and then this is the mobile phase which we have taken in the dropping funnel. So you can do it manually, or just you can by this help of this stopcock here, you can set how the flow of the mobile phase should be. So that flow with how many drops it has to fall on into the column. So all that you can set it here itself, or else you can do it manually by taking mobile phase in a beaker and then keep on adding from the top. So this is the setup for column chromatography. So keep on collecting the various fractions in the conical flask, and then we have to run the TLC. Now what happens when the column is not packed properly? So as we have said that the packing of column is a very important part. Now when we do not pack the column properly, what happens? Now in the first diagram you can see here it is written as uneven packing. So uneven packing means what? See here, the level of this silica is not even. It should be actually like this. This is a uniform packing of the column, but here you can see it is not in a straight way. The layer is bent. Okay, so when you fill the column in this way and the separation is taking place, you will find bands like this. Okay, that means you cannot properly collect those samples which is coming out from the column. So this is the glass wool, sand, okay, and this is the stationary phase. So when the stationary phase is not uniformly packed, then the separation would be like this. So when the separation is like this, the components cannot be separated easily. Then the next type you can see cracks or channels. So these are the cracks. This is the silica which is not being moistened by the solvent. So these are some of the cracks that may appear in the column. So when these cracks appear, then also there won't be any proper separation. So you can see this band which is there, yellow band, it is not properly being separated. Then below the taper, that means when you add the sand on the glass wool, sometimes what happens is while filling the column, some of the sand will go, that is, it will get drained out. Okay, so like this. So when it is in this way, again the separation at the end, even though when the bands are being properly separated on the stationary phase, at the bottom when it is coming out or when it is eluting out of the column, it will be in this way. That means you cannot again exactly collect the pure form of the component. Then when it is dilute or overloaded, so wide bands. So when you load the sample, which is very much diluted, then you cannot easily find the proper separation of the components. You can find like this wide bands are observed. So this is also a not correct procedure for column chromatography technique. So you have to take lot of care while filling the column. Then now the last part, that is we'll just check out the applications of column chromatography. So column chromatography is used to isolate the active ingredients. If a mixture of compounds is given, so which ingredient is active, that you can easily isolate by column chromatography. It is very helpful in separation of geometrical isomers. So geometrical isomers, that is if we are taking an example of cis and trans, we know that in cis, both the identical groups are on the same side with respect to the double bond, and if it is trans, they are on the opposite side. So what happens is when you are taking cis and trans isomers, and if we want to separate by column method, then it depends upon the adsorption affinity between the isomers and the stationary phase. Some of the isomers, if a trans isomer is more adsorbed onto the stationary phase, then the first eluted out isomer will be cis. Suppose sometimes cis isomer may have strong adsorption towards the stationary phase, then trans isomer will be eluted out first. So based on the affinity between the isomers and the stationary phase, the separation can be easily done. In the separation of diastereomers, here also the same case. So sometimes we can have one of the optically active diastereomer. Among the diastereomers, one may be optically active. So that compound may have different affinity towards the stationary phase. So based on the difference in the affinity towards the stationary phase, again, the separation can be easily done. In the separation of tautomeric mixtures. So tautomers, suppose if you are taking an example of keto and enol tautomers. So keto, enol. So again, they have different functional groups. So they will also show different affinity towards the stationary phase. So again, depending upon the difference in the affinity towards the stationary phase, they can be separated into keto and enol forms. It is used to determine the drug estimation from drug formulations. So you can suppose a drug formulation is given, and now if you want to estimate the amount of drug which is present in it, so you can perform this column chromatography, and whatever the eluted drug which is eluted out from the column, again we have to estimate it by some other methods. It is used to remove impurities. That is, if there are any impurities which are present in food material or any that is drug item, you can easily remove. It is used for the isolation of metabolites from biological fluids. So metabolites which are present in biological fluids can also be isolated. So everything is nothing but based on the principle of adsorption and partition. So this is what all about column chromatography, the principle, different types of column chromatography. Then we have seen what are the different types of packing the column, that is dry packing method, wet packing method, and what are the criteria that we have to keep in mind while selecting the solvent as well as the adsorbent, and what is the technique behind the filling of the column and how the column has to be run. And lastly, we have seen the applications also. So hope you have understood this topic. Thank you.